Introduction. Polymerase chain reaction, PCR. The significance of PCR in molecular biology, genetics, and various applications. What is PCR? A molecular biology technique used to amplify DNA sequences. Mention its importance in research, medicine, and forensics. 3. Components required. 4. DNA template, original DNA sequence to be amplified. 5. Primers, short DNA sequences that flank the target region. 6. DNA polymerase, enzyme responsible for synthesizing new DNA strands. 7. Nucleotides, building blocks for DNA synthesis. 8. Thermal cycler, instrument to control temperature cycles. Steps of PCR. Step 1. Denaturation. 1. DNA template is heated to around 94–98 degrees Celsius. 2. Hydrogen bonds between DNA strands break, resulting in 2 single strands. Step 2. Annealing. 1. Temperature is lowered to around 50–65 degrees Celsius. 2. Primers bind, anneal, to complementary sequences on the single-stranded DNA. 3. This provides a starting point for DNA synthesis. Step 3. Extension. 1. Temperature is raised to around 72 degrees Celsius. 2. DNA polymerase adds nucleotides to the 3' end of the primers, extending the DNA strand. 3. New DNA strands are synthesized. Cycling and amplification. 1. Steps 1–3 are repeated in a cycle, typically 20–40 times. 2. Each cycle doubles the quantity of DNA, leading to exponential amplification. 3. The resulting DNA fragments are identical to the target sequence. Applications. 1. DNA cloning. Generate large amounts of DNA for further analysis. 2. Gene expression analysis. Study gene activity levels. 3. DNA fingerprinting. Identify individuals based on unique DNA patterns. 4. Diagnostic techniques. Detect pathogens or genetic disorders. 5. Forensic analysis. Analyze small DNA samples from crime scenes. 1. Recap the key steps of PCR: denaturation, annealing, and extension.